I guess we're live. so much for the video that was supposed to play. I guess I was supposed to hit a button. Or did it play? Did the video play? It didn't play. Well, all right. Well, welcome everybody. Joe Farrar, Dr. Joe from Kingdom Pythons. Glad you all are on. I don't know how, can I tell how many people are on? Yeah, I'm new at this. What are we going to do today? We're going to talk about genetic testing. We've got Tommy from Inky Clouds and Charlie from Gray Rider in the back room, ready to come on, introduce themselves. We've got Dr. Hannah Seidel is going to join us, and there might be some other people that pop on that want to share. So please make sure that you are putting questions in the comment section. I want to make sure that we answer as many of those as we can. We're not going to go deep into genetics. There might be a question or two that you ask that are very deep. And so we can answer that maybe or maybe not. The flow, I'm going to start after we do introductions. I'm just going to start talking about why we started doing it in the classroom. You know, what was the driver for us to do it in the classroom. And then we're going to get into some slides about our process and where we got our protocols from, and just kind of how, you know, we do it in the classroom, how we've done it in the classroom so far. And so that should kind of give you an idea. Hey, if I wanted to get into this and maybe just do it for fun, what kind of materials do I need? What do I have to purchase? What skills do I need? Which is probably one of the biggest things that you have to be aware of, is just that skill base of getting everything where it needs to get. But otherwise, it's pretty simple what we do in the classroom. And then Charlie and Dr. Seidel will probably get you into a lot deeper than you might even want to get into. But that's your choice, all right? So got Mike right now on. I think that's it. So we're going to go ahead, and I'm going to bring Tommy on first.
Hello.
Hey, Tommy.
Hey, how you doing?
You want to go ahead and introduce yourself? And
yeah, I'm Tommy. I have Inky Cloud ball pythons. I'm a hobbyist / breeder. I started looking into the possibility of doing genetic testing for my own collection, because I do have a lot of things that will be producing possets. So I figured it might be a fun thing to try. Then I found out that it's a lot harder than I thought it was going to be. So, and harder to find information on. So I was really excited to start getting to talk to everybody who is doing it and learning more as I went along.
Cool. I'm trying to get on my phone here and kind of see the comments, and so making sure everything's up. Yeah. Well, great. Yeah. And so that's, basically, I mean, we were talking, back and forth. I had posted some stuff after the class had finished their first round of this testing. And I was like, just ecstatic, because I wasn't expecting for them to get, some of the results that we had. And then so Tommy reached out. I forget how that happened, but then one day I think I just said, hey, what about
why don't we try this? Why don't we try that? I'm like, so, yeah, sure, let's do it.
Let's do it. And so, because I was getting a lot of questions, I didn't want to put out there that what I was doing in the classroom was something that is either can interfere with potentially what other breeders might be doing this for as part of their gig, part of what they do, or saying, hey, or giving communicating that, hey, this is something that's so easy. Don't spend your money on sending it to whoever. You can do it on your own. That's definitely not what I want to do. So with some of the comments that were made, and some people were like, great, and I was just getting that feeling that I was making it look a little too easy. And it's even though high school kids can do it, so it's not. And so really want to kind of set that up first. And so then, thought about Charlie. And so here's Charlie.
Charlie. Hey, how's it going?
Why don't you introduce yourself?
My name is Charlie. I'm from Gray Rider Reptiles. I am a keeper and breeder of ball pythons. And I currently am doing the genetic testing on the ball python sheds for people who need that service. I found out about it through Hannah Seidel when she was on a few podcasts, and I just did my research for a long time, and finally I got it figured out after a lot of help from people, and so that's what I'm doing. I'm not a scientist at all, I'm a regular guy, and no scientific background, so it can be done.
Yeah. So what's the mindset for you to even think that you wanted to do that? I mean, why not paint a wall or something?
My brain works like I have to know how everything works. I have a computer science degree, and I always build my own things. Like I build CNC machines. I have a CNC side business where I make things and sell them. So I built all my own machines. After seeing one post on how to make a CNC laser, I was like, wow, I could do that. So I did it, and I got better at it. And now, I make really big CNC plasma cutters and things like that. So I always want to try to do things that most people wouldn't try to do. So when I saw that, I was like, at first when I heard about it, I said, there's no way I'm going to be able to do this, but I have to at least, do a little research. And I wind up finding, the equipment, and it was expensive, but not as expensive as I thought. So I said, all right, before I buy all this, let me do a little more research. And I did the research. I was reading all Hannah's papers and watching tons of YouTube videos and letting it sink in a little bit. And then when I got everything and I started doing it, I was like, why isn't this working? I don't get it. I got these primers made. I spent all this money on the equipment. I think I'm doing it right. And then I realized, like, oh, things have to be sequenced. And, there was a lot of trials and wasting a lot of money and wasting a lot of reagents and time, a lot of time that it took to finally get it to the point where every time I do it, it works now.
Right. Well, that's good. And Tommy, what about your mindset? I know you talked just about your collection and what you have stuff coming in, but you could have easily said, I'm going to go to Charlie and I'm just going to, do you do a panel, Charlie?
Yeah.
Okay.
You do a panel.
Okay. Yeah. So why not just do that? And we'll talk about what a panel means.
Yeah, I'm a tinkerer too. I grow mushrooms. I try everything all the time. And so for me, it's rewarding to learn new skills. So and if I'm going to spend money, I'll spend money on a snake or I'll spend money on some other project. So I may as well do something new. Honestly, that's really the mindset for me. Plus, it's like once I have a skill, then it's an ongoing thing that I can continue going back to.
Sure, yeah. And I think that's important. So, maybe the first thing that we've got to look at, I'm the same way.
Yeah.
I had, when I started teaching a long time ago, I just had this feeling that I needed to have, reptiles in my room. I had a lot of different things because when you're a teacher and you start getting animals, then next thing you know it, you have people starting to donate animals and stuff. And so, they buy this, but they don't want it anymore. And so, well, let's give it to Dr. Farrar. And so, and he'll take it. And it's true.
Yeah.
But I had, so I started with an iguana, and then next thing I have 3 iguanas and a huge parrot cage, and I had 3 snakes. And, but then when I left education and, did a lot of work in working with teachers and, really saw that that's part of me really thinking that if you're going to have an impact on kids, it's more than just what's coming out of your mouth. So what are things that we can try? And so when I came, as I was working with teachers for years, and then I came back in the classroom, and what happened? Well, I bought, reptiles, and actually it was my sons who got the first couple. We got a red-footed tortoise and a bearded dragon, and then we got snakes. And of course, when I started looking at snakes a long time ago, I fell in love with piebald. Who doesn't? And
right.
They were $20,000 or something like that. And when I
exactly.
when I was at a show, there's a male for $350, and I was like, oh, I'm getting it. And so, and then now, that's one thing, having snakes is one thing. What was I thinking to get into breeding? I have no clue. I have no clue, but it's like, wow. I was watching who started off with Chris Hardwick? Watching his stuff. Did you guys?
Yeah.
No.
I watched his stuff because it was just crazy. And I was watching it over and over and over again. And he was very meticulous about things, and he had his ideas and his ways of doing it, but he was also very transparent. And then, again, that mindset, is one thing you need to look at if you want to get into this, I guess, just looking at it and saying, yeah, I want to try something new. So,
Yeah.
All right. So what do you think? You guys have anything else to say before we get into these little slides that I have? Got to have the teacher slides. So get your coffee.
May as well get into the coffee slides.
All right, get into the coffee slides. So this is not a genetics lesson. What this is just some background about what what we did in my classroom. Just so, because people are asking, what'd you do? Oh, that's great. Blah 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 blah. Are you paying? Can we pay you to do it? And I'm like, no. So, so basically, it started out. I was watching Brian's podcast, and that's when I first saw Hannah on there, and of course had me just hooked because of the genetics part of it, and I'm thinking initially collaboration with her to try to set up a way that we can get this protocol in a classroom specifically for high school teachers. Okay, so that's our whole goal. And she was loving that. And she's like, yeah, well, maybe we can write an article and put it in American Biology Teacher or something like that. And I'm like, yeah, that's great. Published work, that'd be awesome, especially for the kiddos. And I just got a biotechnology class, and biotechnology is basically all the skills that you're going to use in this process and then some. And so I had the perfect guinea pig class to do it. And I'm going to start off with a slide that basically just shows you where, where we're at. So I'm going to bring this in, and I think it's going to get everybody else's heads off there. So let's see. Oh no, you're all on there. Perfect. Is that big enough? Can you see that? Mm-hmm. Should I get rid of you guys?
Yeah, that's good.
All right, good. All right, so this is just you can't read this. I mean, you can, but you have to get really close. It's not good for you. I'll show you the individual parts later, but this is the result. So I wanted to start with the end in mind. We had basically, kids who had a research question, and we had snakes. This is our first year breeding. We had Pos het lavender albino snakes. And so as soon as I heard about what was going on and the possibility of, taking this lab-based, university-based protocol and turning it into a high school protocol, this is what I was thinking of. And so, we had already talked about in my class about the snakes. I use the snakes whenever I can, especially when it's about wait a second. We have Hannah's coming on, so let me bring her on. Okay. Hey.
Hi.
How are you?
Good. You can hear me and see me okay?
I can hear you and see you. Fantastic.
Okay,
so
great.
We're glad you're here. we just kind of just give you a background where we've been. Tommy and Charlie introduced themselves. I don't know if you know Tommy.
I corresponded a little bit, briefly.
Cool.
Yeah, super briefly.
So good.
Yeah, I know her all the time.
Yeah. Oh, she shook her head. Yes. So we do annoy him. All right. So, Hannah, let's let before I get into this, I'm going to just make you bigger. Let's see how I do this.
Okay.
Oh, that's not that. That's not what I want. Yeah, it's me. So let's see. Do I do this? All right. I'll make you bigger. And then you can go ahead and kind of just give an intro of you and what you're doing. And then we'll get in deeper into the other stuff you mentioned in the email and stuff that you'd like to talk about and stuff.
Sure. So I'm a geneticist at Eastern Michigan University, and I started a project a few years ago aimed at identifying the genes and mutations responsible for color morphs in ball pythons. And I started this project as both an educational and a research project. So the research is performed by undergraduate students, mostly as part of a genetics course that students take as part of their biology major or their biochemistry major or other science majors. And the students are learning all the standard techniques they learn in a regular genetics class, like extracting DNA and different techniques to analyze that DNA, but they're doing so in the context of performing real research on ball pythons. And so we collect data, and when we have enough data for publication, we publish that paper in a publicly available scientific journal so that everyone can benefit from this research, and so that anyone who wants to use that research to perform genetic testing can do so. And so we've been working our way through different color morphs. So we started with albino toffee candy, lavender albino, and ultramel. We've published that work. We are very close to publishing the paper on the yellow belly complex. So yellow belly, spectre, spark, asphalt, and gravel. And then we are very close now to making a preprint publication available for ghost, which is also known as hypo, and turns out to be the same as orange ghost. Yep. And then coming up, we're just gonna kind of continue working on more morphs. There's another group that has worked on the piebald morph, and so we also have a genetic test for piebald because we've used their data, and we want to make all of our results as accessible to everyone as possible. So for example, Joe contacted me and asked if he could do this in a high school classroom, and so I provided him with some information, and he was doing that. And then other people have contacted me asking me to help them with mini PCR and doing some testing on their own. So I've been trying to help people, though my time is limited in how many emails I can answer. But the mini PCR people just sent me one of their apparatuses, and I've got a student who's going to maybe make some videos showing people how to use it.
That's awesome. Yeah.
That's
and
great.
So there's a lot of content there that we're going to get into as well. I'm not going to get into it. I'm going to basically the flow is I'm going to kind of go into how I started with it. And we kind of went and talked about mindset and just kind of checking ourselves to make sure that, if you want to get into this, doing this as a breeder, there is a mindset that you kind of need to have because, and hopefully we can kind of tease that out, what that looks like, as well as the skill and content and the money in order to get into this. So I just wanted to show them initially just some slides of how what we did in the classroom and why we did it. And so that's where you came in. So I'm going to go ahead and put this back up. So this is actually the first slide. Anyway. Oh, is it still up? Wait a second. Maybe I got to do this.
There
Can you
it
see
is.
it?
We're kind of on top of it, though.
Oh, okay. Let me do this. Does that work?
Oh.
No.
Put us back on the side like you had it the first
This
time.
one? This one? No. It's one of these buttons. It's not happening. All right, wait a second. I had it perfect. What happened to it? There we go. Woo! Boy.
Now we're the Brady
All right.
Bunch.
Cool. Creative Reptiles is on. Hey, guys.
Yeah.
Try looking at everybody else. All right. So yeah, I wanted to start with the end in mind here, just kind of just showing, the reason why we went into this. This is the research poster, just kind of a quick research poster that the kiddos kind of put together. And so the purpose here is, our first breeding season, we had, we put a het lav, pinstripe, and what else was it? It was 100% het lav, pinstripe. I think that's all it was. And then we put that together with a granite yellow belly. Okay. It was our first year. We only had a few females that could go and a male that was ready to go. And we got that from Justin. And so we had these babies, beautiful babies, right? But they were all possets. And so then I get, I see Hannah on the first, I don't know if that was the first show that you were on with Brian from the Snakeatorium, but as soon as I saw that video, I'm thinking, all right, we need to do that. And then hence I reached out to her. So I kind of want to just go through some of the things, and I'll get into what this looks like a little bit, and then we'll talk a little bit about genetics, and then if we got to go deeper because of some questions, we can go a little bit deeper. Okay, but basically, the kiddos had to. They all selected their snake, so we had. Of course, a variety, and I only put this snake up there, but we had a variety of morphs that were either yellow belly and granite. Some took on the pinstripe. Of course, some took on cypress. Oh, that's what it was. It was a cypress pinstripe. And so, I showed this one because it's just my favorite snake, because now we know that it is 100%. Well, we think we know. I mean, based on what we did in class, we still would prove it out. But it's we think it's 100% het pie for our het lab for what we did. So they all picked their own snake, and then we'd been collecting sheds anyway for a while, and so and we've been keeping them clean, and we followed all the same protocols that Hannah has shared many times when she was looking for sheds, and also what they do in the classroom in her lab. So the first thing how we did it was we had them dried and clean, and actually before this we switched over to shredded paper for bedding, and it was a lot less dust, and so we actually used that as a kind of a variable in our testing. So a couple of the snakes that we had were on just a paper, like a liner, and then a couple that we had were actually on aspen, and then most of them now are on the shredded paper. And we did that just because of dust and things like that, and contaminations, and what was going to be easier to keep the sheds. I wasn't even thinking about it, some potential implication in the testing itself. But as we were keeping them, we were constantly going back to what was said about keeping the sheds clean. And so that was the biggest thing that we were trying to do there. So we used just some scissors, and of course everything was sanitized. And we used some scissors and cut little pieces of shed. We didn't know how big a piece. We just figured a little piece. And that's what we did with that. So that's that slide there. And then, so there's really three parts to what we did. There's extraction, and then PCR, and then we did gel electrophoresis. With lavender albino, you can do gel electrophoresis and get your results. There are other morphs that you can't do that. And so that's one thing you need to know. And we were able to do this because this was accessible to us in the classroom because of the equipment we had. I was already teaching my kids about extraction. As a matter of fact, we extracted DNA from salmon testes, which they loved it. And then we did, we did a lot of other things. We did gels, and then we of course did PCR. And so they had the skills down. So this is actually using an extraction buffer, extraction solution that you need, because that DNA is inside the cell, inside the nucleus, and you got to kind of get that out if you're going to do anything with it, right? That's basically the simple, the simple part of DNA that we'll talk about. I just put this here because you got to understand that when we're taking out the DNA, there's sequences there. I kind of use the example of kind of like a recipe book. And so, the DNA is your genome is basically like a recipe book, right? And so every chapter is a different section of the book, but every recipe is really the gene. It's a sequence of these A, T, G's, and C's. And so when you think about it, when it's the master book, it might be your grandmother's recipe book. And so when you go, you got to make a copy of that. She's not going to let you take it with her. So you make a copy of that. And in that copying and in that process, a lot of stuff can happen. But when you get that recipe home, you might change it, right? You might say, well, instead of using 2% milk, I'm going to use 1% milk, or I'm going to use whole milk, or whatever. I'm going to use 3 cups of sugar instead of 2. You're changing what's happening. You've changed that sequence. You changed that recipe. And so just trying to understand when you're taking that DNA out, there's those sequences there, those letters are basically what's causing each of those morphs to happen. So we're looking for a sequence that's specifically about lavender albino. And there are things that you use to highlight that, to make sure that when you're checking to see if it is lavender albino, you're basically using these sequences. So when they talk about sequencing, this is what they're talking about. They're talking about finding what combination of these base pairs are really driving the mutation that is lavender albino. And you know, in a simplistic way, that's all I'm going to say. We can talk about it deeper later, but that's pretty much what you do. So once you get that DNA out and you have it, there are these things that you have to make a solution. It's called a master mix, and in it there's a lot of different things. One of them are primers, and primers are kind of like these flags that go on the front end of a sequence and the back end of a sequence, and then even on potentially the sequence of the mutation. And so what that's for is kind of to tell this machinery, this enzyme, it's enzyme kind of complex, to kind of come along and start basically making copies of that sequence. Because when you take the DNA out of the shed skin, there's not enough for us to kind of see what's happening there. So PCR amplifies that DNA. So it makes a lot of copies, billions of copies of the DNA. And so what you're seeing there is we use Mini 1. We also are using it on Biorad as well, electrophoresis. I show my students a lot of different options. I don't have Mini PCR, but it's similar to this. Okay. And so what you do is you know, there's a recipe. That you have to program the machine, and it's a thermal cycler. So you've got to make sure that you're using the right amount of temperature for the right amount of cycles. And so you get the most copies of DNA that you can get. Once you have all those copies, then you can then run a gel electrophoresis, which I think technique wise is the hardest thing to do, especially if you're using a Mini PCR, Mini 1, because it's just smaller. My students had a lot easier time doing it with a Biorad, a bigger gel. And so, but this is basically a gel electrophoresis using a Mini 1 piece of equipment. It has a hood on top that allows you to put your phone on it, and you can actually do a time lapse of the runs. And so basically what's happening here is you're loading the can you see my cursor? You can't see the cursor? Okay. So these little green things on top that our fingers pointing to, those are just the wells that you put those PCR samples in. And then you're basically looking for, when you run a current through this gel, these base pairs kind of move down the gel. And then based on the length of the sequence, you get a bar at a certain length, and then you can use that to say this is that gene. Okay, in a nutshell. We can talk about more specifics here. So that's what they're doing there. And so these are the gels that we received, and we've got this beautiful image here from Hannah. This was helpful. This is kind of like our standard to look at. So we can tell that based on the bars here, we're looking for het lavender. These are the base pairs. One is the actual normal version of it, because in the het version, they would get a copy of the normal gene and they would get a copy of the lavender gene, just one copy. And so to know that it's het, you'd have to see both bars, right? So we would look at ours using a base ladder here and kind of make sure by counting these bars, which you can't really see them on these photos, to see if these were at the same level here. If they were at the same piece, then it's a good chance that it's het lavender. If it's not, good chance that it's not. Okay, so that is it for my slides. That's what we did in class. Let me remove this. Now we can kind of discuss this to see what you think. How is this different than what is happening in Charlie with you? Let's say what are we doing in the classroom, and then maybe how much more do you have to do to kind of hit the expectations of people sending you their sheds?
Pretty much the same thing. For lavender albino, that's what I'm doing, and for hypo as well, that's what I'm doing. The only difference is all the other genes you have to send out to a lab to get sequenced, and the lab has to have the primer that you want them to sequence it. So, the labs have my primers that I tell them on hand, and when I place an order, I say, everything has to be labeled properly, and you send it to them, and they get it, and I'm guessing they put them in a whole nother big batch with other ones, and they do them all at the same time. But you tell them, this is the test I want done. Well, you just tell them pretty much what primer to use. And then in return, it takes 24 hours or less. Usually I get results from them. And then when I get the results back, they come in a chromatograph, which you guys know what it is. It's just like a line chart, I guess you could call it. And you have to then search for the specific sequence, part of the sequence. And within that, you could see if, I don't know, Hannah, you could probably explain it better. Like, so say if it's supposed to have a G in this one location for it to be an albino, and an A for it to be a wild type. If it has the A, you know it's wild type. If it has a G and it's an albino. But if it has both in that same location, it has both copies of the gene in that same location, then you know it's a het. So that's how that works. And in the beginning it was tough and trying to figure all that out. But now it's quick. Copy paste on special software you use to look at it, and you see it, boom, okay, done. It's pretty quick to analyze that. But it's just when you're doing a bunch of them, you have to be very careful and not cross-contaminate. And in the beginning, I didn't realize that. I contaminated everything, and I had to buy all new pipettes because I had to buy all new reagents, everything, because I'm like, why is this not working? And I realized I contaminated something somehow. I have no idea how it happened, but I didn't realize that PCR, when you open up the little tubes that you put in the PCR machine, tiny little invisible particles come out because there's billions and billions of copies of DNA in there. And what do they call it? Like a mist, or I forget.
Master mix?
No, nothing like, it's called like a mist or something, but you don't see it. You can't see it.
Aerosol, maybe is the word you're looking for?
Aerosol. And it just gets on everything. So, the people in mini PCR were great. They were telling me, like, listen, you got to do that in a completely separate room. You have to wear a lab coat. You have to change your gloves. Have 2 sets of equipment in different rooms. And this, so I did. I converted my office in my basement to my PCR part, and then the living room in the basement is where I do my extraction and everything. And I have separate pipettes for each location, separate reagents or anything for each location, gloves in each location, pipette tips in each location, and bleaching everything when you're done. And of course, knock on wood, I haven't had any issues since I started actually doing that.
Yeah, that's another huge element. I mean, I wasn't even expecting to get anything. And I basically, the first run was them trying to work out their own protocol and what safety elements they had to consider. They decided initially first, instead of using the small PCR tubes, they used a bigger tube to put the mix in and stuff. And then they, the extraction and stuff. And then they took that out and they put the master mix in another bigger tube. And then they did all that. And then they aliquoted it into the smaller tubes because they were having problems, taking the lids off and crushing the tubes. And so, you learn these things. And so that's what they were doing. And then, again, it takes.
Yeah, I was doing individual PCR tubes for every single one. And then people in mini PCR were like, no. If you're doing multiples, make one batch. One, you're saving money on the pipette tips because you're not going through them as fast because you have to change them anytime. It's a one-use thing. That's it. And, so at least if you're doing water and everything, it's boop, 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 you could use that. But if you're, doing one at a time, you're using 8 pipette tips instead of 1.
So we have a question about, how much does it cost to have your own small lab?
$1 million, maybe. It seemed like that, but no, like Mini One, I found them too after. I don't have anything from them, but Mini PCR, I mean, you could get a whole kit for, I want to say it's like $2,000 or something, maybe. I forget how much I spent, but then you get the upgrades. And I feel like the most expensive part was the learning part, blowing through reagents, blowing through primers, paying for shipping on all this stuff, because you're just not doing it right in the beginning and you're just wasting a lot. That definitely was an expensive part.
Yeah, for Mini One, I just looked it up. It's about $1,200 for the PCR and the electrophoresis package, and that comes with a pipette and tips and things like that. Of course, you've got all those other reagents. If you're a teacher, Hannah turned me on to some great courses that were
right.
free.
And then you're going to want this Mini
Right.
PCR too.
Exactly.
Centrifuge. I also bought one of these, which I didn't have for the longest time. Vortexer. This thing is amazing.
Yeah.
Like, to mix stuff, I feel like it's helping me so much. Just making sure, like, especially when you have the primers just sit. I don't know how it works, if things separate after they're sitting for a while or what, but I just mix everything on that before I do it.
Mhm.
And it just makes me more comfortable that everything is getting combined well.
So Hannah, what do you think?
Well, one update I have is that we are trying to make some of the tests easier. So Charlie, you mentioned that some of the tests like lavender albino and hypo, you can do a PCR and then run it on a gel and get your answer. And for other tests, you have to send it out for sequencing, which is just another few steps. So we're trying to revise some of the tests that currently require sequencing to allow them to be run like the lavender albino or the hypo, because it's easier and faster, cheaper. So I've got a student whose project is to basically do that for as many of the existing tests as we can. So we're starting with piebald and yellow belly because those tests are the most common requests that we get.
Well, yellow belly, you don't sequence, do you? You do the cutting with the.
So you can either sequence or you can cut with the restriction enzyme and then run it on a gel. So we're trying to revise it so that you don't need the restriction enzyme and you instead just run it on a gel like hypo.
That would be great because I hate doing that. It adds a lot of time.
So I think that if our initial design of a test works, we actually should have that in the next few weeks.
Cool.
So then I can share those. It would just be one or two additional primers that would need to be purchased, and then it would be just like all the other tests. It'd be really easy.
Those are the things that have been keeping me up at night doing research, trying to figure out how I could use your information to make my own test to run on a gel. But if you guys are doing that, I'll let the pros do it.
I mean, I can't promise it's going to work for every test that we have. It has to do with the type of change in the DNA. Some are easier to detect than others. The yellow bellies, I think, will be pretty easy to detect. I'm less positive about the piebald, but I really won't know until we try it.
Cool.
Yeah, that's what I was going to ask. What is the post-processing? Like, what's the determinant that says that you have to do an extra step after the gel? So you answered that.
Yeah, so some of the changes in the DNA are deletions, like they're big chunks of the DNA that are just missing from the genome. And those are easier to detect, whereas other ones are just a change in one letter to another letter, and those are more subtle and harder to detect.
Gotcha.
Is that because on the gel, the base pair, the size is just not that different? So it won't be that much of a change in the gel because the size
it won't drop further or whatever.
It'll be right there.
Yeah, so the piece of DNA on the gel is going to run at the same size, even if it's a different letter. So if you change just a G to a C, for example, as long as the piece of DNA is the same number of letters, it will look the same to you on the gel.
Yeah, so the bar looks the same. It goes to the same weight.
Yes.
Gotcha.
Exactly.
Oh, okay. All right, that makes sense.
And then the mini PCR people are interested in selling a lavender albino kit, like specific for that type of test. And I think they might be interested in selling any kit that we can make that is just a PCR and then gel. So that would be hypo and then these others that I've mentioned. So that could simplify things for people a little bit because they could order everything from one company as opposed to ordering the primers separate from the mini PCR equipment.
Yeah, they reached out to me today actually for something else, but I've been talking to them about that, and I think they're going to send me to be their guinea pig since I'm not a scientist, to make sure that I could do it.
Do any of these changes, like for example, ditching the restriction enzyme, do any of those changes cause any, concern about reliability or other issues?
Oh, that's a good question, actually. So, I would say that the way that we're trying to redesign it would, if anything, make it more reliable because it would reduce the restriction digest step, which means that it reduces a step in which you could make a mistake. So by making it simpler, you're often making it more reliable because you're eliminating a place where someone could make a mistake.
I have to teach about restriction enzymes anyway, so I might as well.
Continue doing it.
I got it. Well, I'm not doing it, but I need to figure out how to do it.
Oh, gotcha.
That's good. So here's a question, just as far as any process documentation available for those already doing it. Well, I'm going to post what I do in the classroom. But that's just for laboratory albino. And most of that information comes from Dr. Seidel. So, how we've changed it. We're still working on the PCR cycling and it being 96 minutes long and kind of trying different mixes and stuff. So
mine is like almost 2 hours. I don't know. Yeah.
We use the fast tack, I think, on that one.
See, I don't know enough about that. I have different tacks from different companies, but the thing I've had success with is the mini PCR 5X master mix. Mm-hmm. So I kind of don't want to change it until I master something else.
Yeah, sure. Yeah.
But I know it can't be that much different. I just don't want to screw anything up. So I'll test my own stuff and on the side.
I think for Joe, the time is an issue because he's doing it in a classroom setting. So all that time, when you're teaching, that really makes a difference. If you're just doing it for yourself at home, the time isn't really a big issue because you're not doing something for those 96 minutes. You just set it up and then, you can come back the next day. So that's kind of downtime in terms of the human working
One thing I do during the PCR process is about an hour in, I always have a timer go off. That's when I go make my gel. So after it goes in, I know in an hour I go make my gel so that's ready to go. I take my ladder out of the freezer, so that's ready to go. Those were things I did not do in the beginning, and then I finally realized I make a step-by-step to the freaking second, like what I have to do, because I would forget. Oh, I didn't take the ladder out, or I didn't take this out. Now I've done it so much that I don't need to look at anything, but it's in my head. All those steps, like, all right, after this you do this, after this you do this, to keep the.
So what, Charlie? What's the process for making the gels with mini PCR?
So in the beginning, I was just using they have literally tablets, and it comes. You just put distilled water. I think it's to make a 2% gel. It's 20 milliliters of water with one tablet. Mix it up. You dissolve it. Put it in the microwave. Boil it for a little bit, and then you pour it into. Do I have that here? I didn't bring that up here.
The little box with the comb.
A little box, and then you put the comb in there to make the wells. I think it probably takes 15, 20 minutes, and it's ready to go. But now to save money, I bought the dye itself, and I just used the Aragos gel tablets.
Sure.
Because the ones with the dye in it and everything are a lot more than just using Aragos gel, putting one you will dye in it when you're done. So that's saving me some money doing it that way.
Yeah, Mini One has these little gel cups. You put them in for 15 seconds, 20 seconds, and then you just pour it into the tray.
So you don't have to microwave or anything?
Yeah, you microwave it for 15, 20 seconds.
Oh, okay, yeah.
And then you just pour it in. You can make one gel or two gels. It's pretty neat, pretty easy. I don't like to use it because I want them to make their own gels. But for this one, we used the cups, so it was actually worked really well. All right, so there was another question.
I will say one thing in the beginning, like obviously I don't have the background, and I read all Hannah's stuff on her website and her papers, and I'm like, okay, this is the primers you use. And I figure, okay, so this is what a primer means. I thought I was going to be able to use the gel for everything. I did not know that sequence with this primer meant I had to send it to a lab. And that was after I spent all the money on everything. I'm like, all this money, I can only imagine how much it's going to cost for me to get these things sequenced. Then I started looking, I was going to buy my own Mini One sequencer. And I was like, all right, it's not that expensive, like a couple $1,000s. But then I realized how much the, what are the cartridges called? The
Lanes, maybe?
Yeah. They only last a certain amount of time. What's it called? For the sequence machine. I forget what it is. I'll find it. But they're like $900 a piece. So unless you're doing tons and tons and tons of them, it wasn't worth it for me to buy that machine. But sending them out isn't that bad. But do know that there is more than just running it on a gel, because I did not know that when I started.
Yeah, I didn't. I mean, as far as the primer, I knew about primers and things, but I was hung up on the third primer, and I went back and forth. But you just kind of get some lesson on that, because I just always, for some reason, just thought about the forward and reverse, and that was very interesting. We actually did more research on that. It was a great addition to the class. So. That's you learn these things, and that's the process of science. Screwing up is important in science. So
the flow cell, that's what I was thinking of. The flow cell. You put a flow cell in the sequencer, and if you have to reuse it, you have to clean it out, but you could only do that a certain amount of times. They have shelf life, and those flow cells cost $900 a piece. So
is that the similar in all the models out there? Have you looked at other models?
Yes, yeah. Everything I've seen, you need a flow cell in it. I'm sure there's other ways, but those machines I think are very expensive. Hannah?
Yeah. I think the type of sequencing you're talking about there with the flow cell is a different type of sequencing than when you're sending out just individual samples. I feel like if you're just testing an individual animal for something, the definitely the most cost-effective thing is to do what you're currently doing, which is to send out those individual samples.
Okay. Yeah.
Cool.
Well, awesome. Sequencer.
And like I said, I'm hoping so essentially what I have right now is a student who is needs a thesis project. So I've made her thesis project to try to develop all the current tests into tests that just require a PCR and a gel to make things easier for people doing them on their own.
Mm-hmm.
That's cool. What else do you have coming up? You also have some. You were looking for some maybe pictures, or the ones that I sent you?
Yeah. So one of the things I still need for my ghost hypo publication is I need a photo of a single gene ghost hypo animal, and it needs to be an adult, or at least not a hatchling, and somebody who's willing to give me permission to include it in a scientific publication. So if anybody has a photo of that type of animal that they would be willing to contribute to a publication, I would credit the person in the caption of the photo, so they would get credit for the photo. I'd love to have a photo of that.
Single gene, you said?
Yes, it could be an animal that's het for something recessive, but as long as the only thing that you can see in the animal is the ghost, the hypo, then it's fine.
I've got that.
Okay, great.
I'll show you over to you. Limey, we're not letting Limey do it.
All right, take him up. I'll hide him.
Bye, Limey.
Should
That's what he gets for being mean to me.
Maybe I can suspend him for a second.
No.
I'm joking.
Goodbye, Limey. Yeah, there's mostly people who are asking questions about upcoming tests. I haven't seen like Desert Ghost, Puzzle, What's Next, Ultramel.
Ultramel,
like
there already is a test for. So Ultramel is a little bit complicated because there are two different mutations for Ultramel. Some Ultramels have one mutation, some Ultramels have the other, and some Ultramels have both. We currently have a test for one of those, and we are basically very close to a test for the second one. So then we'll be able to identify both Ultramels. Somebody sent me a monarch recently basically for me to test, is Monarch the same as Ultramel? And as soon as we get that second Ultramel test, then we'll know for sure whether Monarch is or isn't the same as Ultramel. I know that there's another research group working on finding morphs. There's lots of people who have asked about a clown test. That seems to be the most popular one. I think that other group is working on clown. I purposefully am not working on clown because I know that other group is working on it. And I think Rare Genetics Inc. says that they have a test for clown. They haven't made anything about their test public, so I can't, I don't have that information myself about that test, but I suspect that if this other research group is really working on clown, that will be something that becomes available maybe in a year or so. Yes, I'm looking for more. So what I'd really like is more genetic stripe. Also, anything in the blue-eyed leucistic complex, especially the less common ones. So like bamboo, special, Russo.
Okay.
What else? I would love to have more Suma and mahogany, and also any that are super cinnamon or super black pastel or black pastel cinnamon. Yellowback, so super fire. I'd love to have some more of those. Let's see. Those are the ones that I immediately can think of. I will say that often the limitation of whether I'll say our choice of what morphs to look at, morphs to study, is made in part by what we can get enough samples of. So for example, rainbow would be one that would be nice to study. I think there's some very obvious genes for that, but I only have like 1 or 2 rainbow samples in my collection. So I can't do a lot with just a couple samples.
Yeah, I know people are asking me about rainbow,
and you need visual, obviously, when you're first starting out.
Yeah, so I do not trust het.
Understood.
People describe their animals as het because they're sometimes wrong, is the truth. So,
and as an aside, when we're looking at Monarch and Ultramel, if we're testing and we say Ultramel had 2 different variants, right? How are we knowing that it was not an Ultramel and a Monarch presenting?
Yeah, that's a good question. So when I say that Ultramels have 2 different variants, what I mean is that I've collected, let's say, 15 Ultramel samples. And all those people said, this animal's an Ultramel. They never said, this animal might be a Monarch or an Ultramel. They've just said, here's an Ultramel. And 10 of them have one and 5 of them have the other. And so that's what I mean when I say that there's 2.
That makes sense.
And actually the same is true for albino. There's 2 for albino, and there's also 2 for yellow belly. So it's actually pretty common that there are multiple, which is not surprising as a geneticist, because anytime you mess up the meaning of a gene, you end up typically getting the same color trait as the output. So you can mess up a gene in a few different ways and still get the same color output, which means the animal would look the same to the breeder, but even though the molecular genetics might actually be a little different.
Okay.
Sure. And then how does it, the amount of expression as well, right? I mean, we had a question that I wrote down that someone was asking about that they had 2, let me see what this was. Two of the same morphs, but one had a different, like sometimes it comes up with Desert Ghost. Is it enhancer or not? And so there's a difference, what they think is the same recessive snake, but the expression of that gene it varies.
Yeah, so what you're referring to is basically not every animal that belongs to a morph looks exactly the same. And that is probably due to other mutations elsewhere in the genome. So you can imagine there's one mutation that, say, causes you to be albino, but you could have that, and that is responsible for the absence of the brown and black coloration. You could have other mutations elsewhere in the genome that are responsible for whether the red, orange, yellow areas, whether they look really like faint yellow, or whether they look darker orange. And so both those animals would be albino, but they might not actually look exactly the same, even though they're clearly albino. And so you can kind of extrapolate that to any morph category where there's still some variation within the morph.
Right. And then this another question similar was, is there a way to kind of, or have you noticed a connection between, let's say, the pied markers in a het to the percentage of them being accurate, that you can use those markers.
Oh, do you mean like the ringer, the ringer phenotype?
Yeah.
Is
No,
that?
the tracks on the belly, usually they say if you look
Yeah.
at the tracks
Oh.
on the belly, it's like a magic marker on both sides.
That's a really good question. So I haven't looked at that at all, mostly because people haven't been telling me that information.
Mhm.
I will tell you that we, in our study on yellow belly, we asked how often are people right when they describe their animal as yellow belly. So we collected, I think it was 130 animals that were described by their owners as yellow belly. And they were from, all different people all over the US. And we said, okay, how many of these are right? And the answer was 3 out of 4 times they were actually yellow belly, and 1 out of 4 times they were not. And often when one from one owner was wrong, so were all the other ones from that same owner. So I think there is some, there's an owner component there to.
It's
Absolutely.
an onerous component.
So I have a question. What types of things would be if people are interested in either learning about this more or doing it, what types of things could be the most useful for, let's say that I had students who needed projects and they wanted a project that involved science communication, and maybe they were going to make either some documents or some videos. What types of things would be some topics that people might be interested in having a video about or having some more information about?
Yeah, I think based on what's out there already, that I know in the education community, at least in the secondary education community, popular videos about skill, in the context of doing those. I know like Biorad has a lot of those videos out there where they're, if you're talking about P-Glow, doing transformation, something like that, they have, the entire setup there all the way down to, when you put it in ice and all that stuff. And so I think things like that, very not just an overview, but very kind of detailed with maybe chapters in the video, so you can go to certain areas, are beneficial, especially comparing multiple pieces of equipment as well. That's important.
When I was learning and still learning, and every single day I research it, I was on YouTube a lot. There's nothing on YouTube showing you this exact process from start to finish that has to do with reptiles. Or there's tons of lab stuff that you could watch, that a lot of plants, a lot of human things, and a lot of extracting from blood, a lot of extracting from mouse tails, this and that. There's nobody shows how to extract from a shed, or even like a mouth swab, or like, and if you Google PCR, you're just getting tons of things about COVID tests, and it's just, there's so much stuff that is not useful for this. I mean, it is. I've watched hours and hours and hours of videos, so a lot of people have been asking me to start my own YouTube and show a lot of the process and everything like that, and I might, because I think that coming from somebody who's not a scientist, it's going to help other non-scientists. Like I was telling somebody the other day, I'm like, I'd probably be the best teacher for you because I screwed up already. Like I was the idiot that thought I could do it already and made all those mistakes, made all those assumptions, bought the wrong things, did the wrong things. But also it harms you, I feel like, by not going through those. Because I could literally, if somebody came to my house right now and said, this is what you do. It's not brain surgery if somebody holds your hand to do it. I could show anybody how to do this, and the next time they could do it by themselves. But when something goes wrong, why did it go wrong? Not screwing up, kind of hurts you.
Yeah.
I don't know. But
I don't know. I think the hardest part for students of mine is when it comes to loading a gel. That's loading.
That to be the easiest part of everything. Like when I sent my pictures to Mini PCR, they were like, "Oh wow, what loading die are you using?" I'm like, "What the heck's a loading die?" They're like, "You're not using a loading die?" I'm like, "No." They're like, "That's like perfect." I'm like, "I don't know. That came easy to me. The loading the gel was easy."
But it's hard. It's hard, and it's good for you that you are doing it. But I'm just saying, From a teacher's perspective, it's hard. I mean,
Balance it on
they're
there.
gels. That's what I tell them. But they all have their own
And you
little.
can't go too deep, because I did that once. You go too deep and you see like, why are these weird lines at the bottom? Like
you push it down, you
the
put the
bottom
pipe
of the
too fast, and then
All the
you
way
got,
through it. Yeah.
So there's a lot of contamination can happen in that step from your gels, and so that's a difficult task.
So with contamination as a topic, this is not something that you would employ like a stiller box or anything like that for flow hood, blah blah blah.
Are you asking me? I'm
Whoever, anybody.
I'm actually less careful than Charlie is describing
Okay.
he is, and I don't have problems with contamination. So
Okay.
I'm not sure what I'm doing differently.
Yeah.
I do always perform a reaction that's called a negative control reaction, which is a reaction that contains no DNA, and that allows me to double check that I don't have contamination.
Yeah, I found something about that. It might have been in one of your write-ups that where it's like, hey, make sure that you have this as kind of like your checks and balances here.
Yeah, so what I tell my students is that PCR is really, really sensitive. So it can amplify a teeny, teeny, tiny amount of starting material. So for example, people who are employed in the forensic labs that analyze crime samples, so let's say a sample from, some type of murder or rape or something like that, and they have some tiny amount of DNA that they want to analyze, they have to be really careful about contamination because they can end up amplifying the employee's DNA. And actually, when people work for those labs, they often have to provide a DNA sample from themselves when they first start working there, because otherwise their own DNA could come up as matching some crime scene sample. And this is true, like with the snakes. So that's why it's really important to be careful about. I am really careful about making sure I don't have like stray pieces of shed around. I only handle one shed at a time. It comes out of a bag. I put it into a little bit into a tube, and then everything goes back in that bag, and I put it away before I get the next shed out, because I don't want any movement of little pieces of skin from one thing to the next.
Now I have a question that I'm always wondering: can my skin cells and things like that affect, because the primers we use, would those, say I took my own DNA, extracted it, would those primers have anything to attach on?
No, they wouldn't. Your DNA is different enough from a snake and for this particular gene, it's different enough that they wouldn't. The thing that you could get from yourself is you might be able to introduce some contaminants that might inhibit the biochemical reaction, but they wouldn't cause a band to be observed for you.
I figured that, but because I think that's why I'm extra careful, is because I always not sure, can my DNA screw this up? And I'm like, I don't think it can, but it's good to know.
I had another question here. These again, just people that sent me stuff. So when you're developing the tests, what do you consider good accuracy rate, before it becomes something that and I guess Charlie, that's for you too. This was specifically in, if I'm going to spend the money, is it 99% accurate? Is that the kind of level of accuracy you're going for to get a positive result?
You're asking me, for example?
Either you or Charlie for doing this. It's just a question that
I'm not the one who's the shoot. They're the one. I use her data, so I haven't made my own test yet. So
yeah. So I guess I would say that the tests. So typically with these reactions, either the test kind of works or it doesn't. So the accuracy. So okay, like this piebald test, for example, that we want to revise the piebald test so that it's easier. So what I would probably do is design new primers that would allow this test to be easier. I would test those primers on one piebald animal and one not piebald animal, just to see, do they even work? If they don't work in those two animals, then I throw them out. I need to redesign. If they do work in those animals, then I need to confirm that they really work in a bigger variety of animals. So I might choose 10 piebald animals and 10 het piebald animals that I know using my old test are actually het, because I don't believe people when they say their animal was het piebald, because sometimes they're wrong. So I would choose 10 of each, and then I would run them. And if they all came out exactly the way that I expected, I would say that that's good enough.
So you have your own animals now?
I know I don't have my own animals, but I have samples. I have like maybe 10 sheds that people have sent me that they described as het piebald that I have tested in my old test, and I know for sure are actually het. I would use those as my standard of het piebald.
So have you considered collecting or keeping?
Uh-oh.
You're too busy for that.
I have really I'm so busy I can't even answer emails. So I feel like I'm not ready to have a live snake at this point.
I'm joking. So the last question that I have, I don't know.
There were some comments in the chat asking if I needed monsoon, and yes, please.
Yeah, I responded, and then they asked if you needed.
Because I'm skeptical. I've been burned before on hets when someone says it's het and it's not. So now I no longer am. I don't always feel confident when someone describes something as a het. So really, to study a morph, I don't use hets as my first chunk of experiments.
That's fair. That makes sense.
Yeah, that's. I mean, I do that just because of just purchasing, and I don't want to waste money. So we try to do everything here in the building, in my classroom with if we produce the het, then we're going to put it to a visual, just because we don't have that much time and money to spend on mistakes like that. All right, this one question was, we'll do this one first, and then go to this last question that was sent to me. What steps should you take to prepare sheds? Like, so for example, we just kept them clean in a bag, the way you described it on a couple of shows. But is there anything else that you can do to prep the shed for the extraction, and then whatever else happens?
I have found that the sheds are stable for years as long as they're dry, and the dry is really key. Because sometimes people send us sheds that I know were wet and spent a long time wet, because they're like these sort of clumped up block of gook. And those, I really have trouble getting DNA out of those. So basically the key is get it dry as soon as possible and then keep it dry. And then it should be good for quite a long time.
And not in the freezer, right? You can just keep those out?
You can just keep those out. I do sometimes get sheds that are infested with insect larvae, and I put those in the freezer to kill the insect larvae. But if the shed's not infested with anything, it doesn't need to be in the freezer.
Okay. So I've had filthy sheds come to me, perfectly clean sheds come to me, wet sheds come, and I say I probably extracted DNA from a couple hundred sheds at this point, and out of that, 3 or 4. I have not been able to get DNA out of, and I have no idea why. I've redone it, tried different things. There's no rhyme or reason to it. I thought, like, I just had one the other day, and I said, "Hey, how old is this shed?" He's like a week. And then I've gotten DNA from sheds that somebody's like, "This is 6 months old." I don't know. I don't know why some of them I can't get DNA from. But do you have any idea, Hannah?
I don't know. I've had a similar experience where I'll get a really low yield of DNA out of a shed, and then I'll think maybe the student made a mistake, so I'll go back to the shed and get another piece, and I get the same thing, like very low. So it's something about individual sheds, and for sure the wet ones for me, I have that. I've also had that for sheds very rarely that perhaps weren't wet. It could be something about, like, I suspect it's not that there isn't DNA there. I suspect it's something about maybe like the media or something that the snake was in that is coming through the DNA prep and is actually inhibiting the reaction. But that's just a hypothesis. And again, it's uncommon for that to happen. So although it can happen, it's really not the norm.
So I've done 2 things also. I had somebody send me a swab of a mouth. I've done it myself, and that works. So I had somebody send me, I'm in New York, they swabbed their snake's mouth. They're in California, they shipped it to me, and I got DNA out of that. And it was perfect. And then somebody recently asked me if I could do scale clippings and sperm plugs. So if you have a male, and you can pop it and get a sperm plug and ship it. So Wednesday, I did it to one of mine, and it worked. I got DNA out of sperm plug.
Isn't the sperm plug a shed anyway? Like it's not really
Whatever
a
it
sperm
is,
plug.
when you pop it and that white stuff comes, it was gooey and not.
Yeah, it's like a hemipenal shed.
Yeah.
What do you think there'd be stuff from the female there too? Or am I misunderstanding the sperm plug?
You have to pop it out of the male, so it would.
Yeah,
it
it wouldn't have anything with the female.
Okay.
I mean,
Yeah.
unless they
It's
had
kind of like
recent contact,
male
maybe.
sperm, you know.
Okay.
Yeah, unless they locked.
Yeah,
maybe
recently, I
that morning
imagine.
and then that night you did it. You know,
Okay.
it could possibly.
I would think that could be if it was recent, I would think that could be potentially an issue.
Right.
Right.
Yeah, that makes
Agreed.
sense. Mm-hmm. But scale clippings, I haven't tried. Honestly, I don't know how the heck to cut a scale on a snake. So maybe I'll watch some videos of that.
I think it would hurt the snake to do that because the scale it's like a fold of their skin.
Yeah, and what would be the advantage versus just waiting for a shed anyway?
Not waiting.
Yeah, not even
that's silly.
with mouth
If it harmed the animal, then I'm not doing it.
Yeah, it would definitely harm the animal. It's part
like
of
what
part of
type
their
of
skin.
contamination could come from mouth swabs, you know, if they just ate.
I doubt. I think mouth. So we've used mouth swabs as well. It's just like you have to convince the snake to open their mouth, which is, in my experience, is not always easy. Other people maybe have better luck with that. But certainly that would be a very feasible alternative. A mouth swab.
Yeah, I know people that have done it.
And could you so this guy, he used a kit that he got for nido testing. So it was the swab in the plastic and sent it to me. If somebody doesn't have that, could they use a Q-tip and just throw it in a Ziploc?
Probably.
Because
I would probably tell them to let it dry out
that's
before
what I was
sending
going to
it
say.
to you.
Because I'm an investigator with the state police, and I know when we collect evidence, any wet or liquid evidence goes in paper. You don't, which I'm like, why is that? It's because it could grow mold.
Yes, that's right.
So you don't do that. So, yeah, so swab, let it dry, and put it in, and it should be good.
Cool.
But some people, people are impatient. They don't want to wait.
Yeah.
I couldn't imagine cutting my snake because I didn't want to wait. But
well, I think if you cut a scale, it's permanent. Like that scale.
It's forever.
Return.
Yeah. It's
Yeah.
out of there. Yeah.
That's why I said I'm not
dangerous.
trying that until I look into it, and so.
Yeah, it's all one piece. It'd be like grabbing your elbow and just being like, let me just
it's
put
like
part of
taking
this
a
off,
biopsy, taking a punch biopsy. Yeah.
Yeah, except I don't know that they have the regenerative process. It'd be kind of like it'd be a scar.
Yeah, I think it would.
Yeah.
And I Googled it really quick, and I feel like I saw that people do that to mark and identify snakes.
That may be true. Like on the belly, potentially.
Oh yeah, like a belly clip. They do that. Yeah, they punch part of the scutes out sometimes. Yeah, for like when they're tracking canebrake rattlesnakes and things like that. When I was doing that with a scientist here in Virginia, we would do that if we collected them previously.
Great. All right, I think this is the last question. I'm going through my notes here. So where are we at with sex determination? Do you see a kit coming for sex determination instead of having a popper probe?
I'm not currently working on that. I don't know another group that is. I think that Rare Genetics Inc. might say they have a test for sex, but again, it's not public, so that's not something I can evaluate as a scientist. I would say I don't certainly have it available anytime soon.
I'm quasi working on it. I've been doing a lot of research and talking to a lot of people, so I'm hoping that I could get it at some point.
Yeah, RGI does offer it, I believe, from what I remember.
We'll see in the chat. Nothing else in the chat.
I believe so. Limey saying they have lots of spare scales. That's all I see. Just to recap on the sheds that you do need, we have Monsoon, G-Stripe, Suma, Super Black Pastel, or Cinnamon.
I have
Anything
Super Black
else I can't
Pastel
remember?
for you.
I have that also. But is there anything else anybody else should send you? I can't remember the
mahogany
list.
goes with cinnamon. Oh, and then the less, basically anything in the blue-eyed leucistic complex,
Okay.
but especially the less common ones.
Less common. Okay. Awesome.
All right, cool. Very cool. I have to show my banner. My students, my students will get mad at me. How can you support Kingdom Pythons? How about buying a rat? There's the Venmo.
There you go.
There's your Venmo for a rat.
There's
Can I get a Dumbo rat?
whatever you want.
Okay, yay.
It's your rat. Well, that's it. Yeah. So I've got them working on some entrepreneurial things. Let's see. There's a comment up here. What does it say here?
That's KG.
KG's on. All right. Hey, I got to show that
Yeah.
banner again for him.
So he can buy a rat. KG,
Yeah,
buy a rat.
he needs to see that, KG. You just need to see the banner. Take note. Take a screenshot right there. There you go. All right. Well, thanks so much. I think this was great. I
Yeah,
mean, it was
had a good time.
definitely different than
I just
the
bought a couple rats for you.
Oh,
Yay. There you go. There's some rats.
it was definitely different than a lot of the other conversations that have been out there about genetic testing. I think we provided a lot better, a little bit more information about what it takes to actually do this yourself if you wanted to do it on your kitchen table, and maybe we won't be seeing that happening, but looking forward, Hannah, to the things that you're doing in your lab, and then our next step in our classroom and how we can collaborate possibly on the next thing we're doing. But right now we're having fun with lavender albino. So thank you so much for your help there. And you're going to have a lot of kiddos who are watching. Well, they're actually graduating tomorrow, but they're going to watch the playback in this, and you're a hero to them because they love my feedback on my biotech class was engaging in the work that you've done and watching the podcast and things that you've been a part of. They really enjoyed that collaboration that we've had. So that's been great. And Tommy and Charlie, looking forward to continue stuff with you in any way that we can help you guys. Again, just thanks for being there. So
if
how's
anybody
that
wants
yours
to
doing?
do it on their own or has any questions about how to do it, and don't feel comfortable talking to a scientist or anything.
Oh, you're a scientist now. You can't push it off anymore.
I'm not a scientist. And that's why, I'm using all of Dr. Seidel's stuff. So, I mean, people ask me, no, you could do it too. I'm willing to help anybody and tell them how I do it, and because it's not my research. So,
All right, I'm coming up
grateful
there.
for her. So,
well, I'm posting everybody's info in the description, and I don't know how I do that, but at some point I'll figure it out. I'll try my hardest. All righty. Again, thank you so much, Hannah. How's your son? Right, you had a little baby boy.
Yeah, he's in the other room crying right now.
Oh no!
Go take care.
All right.
He's doing well, but he's not a good sleeper. So
He's not a good sleeper. Well, sorry about that. That'll change.
Yeah.
That'll change. All righty. Well, you guys take it easy. Thanks a lot for being here and willing to do this, and I appreciate you all.
You got it. Take care.
Good night.
Now,
bye. Have a good evening.
Bye. Nice meeting everybody.
Bye.
You too, guys. Bye. All right, everybody. Thanks for showing up, those who did. And if you are on and you'd like to subscribe, go ahead and do that for us, please. That'd be great. Looking at the comments for any last ones. Congratulations on the baby. Are you still working with mini PCR from home testing? I use Mini 1, but I'm going to try mini PCR, and I think Charlie uses mini PCR. I think that's it. More primers. Everybody wants more primers. Yes, we do. All right. Have a great night. Like and subscribe. And we don't do this often, so you'll just have to look out for the next time we try to do this. I'm just trying to get better at this streamcast stuff. So have a great night. Have a great weekend. Ciao.